Hello, my dear students. Today we shall continue our class in which we were dealing with the centromere and segregation fidelity. So we have already dealt with the structure of centromere in the last class, and in the previous class we were dealing with the centromeric specification by the centromeric protein A, which is also called CENPAA. So we have already seen that how CENPA and its variants, which are together called CENH3, they impart centromeric property on a particular stretch of DNA, and how they differ from the canonical H3. So we have seen in detail in the last class, we have seen the structural variation of centromere in different organisms, including the point centromere in the budding yeast, the regional centromere, which was moderately complex in fission yeast, and finally, the complex centromere, which was also called satellite centromere because of the presence of tandemly repeated satellite DNA in case of human centromeres. So the specification of centromere is not only genetic, but it is also epigenetic, and we shall see in today's class that how epigenetic specification of centromere takes place. So first we shall deal with the difference between genetic and epigenetic factors. So let us begin with the slide share. So let us have a backgrounder of genetics, that the flag I have added in my slides, back to the basics. So first you should have an idea that what is epigenetics. Let me adjust the picture a little bit. This thumbnail is coming in between the slide. Now it's okay. Yes, it's okay. So epigenetic factors. So what is epigenetic? Epigenetics, as the word indicates, it is above and over genetics. So the genetic factors are the genetic information in our case is encoded in the DNA, and it is encoded in the DNA as a sequence of nucleotides present on the DNA, that is adenine, thymine, cytosine, and guanine. These nitrogen bases, these are the 4 letters through which the entire volume of our genetic information has been encoded. So ATGC, if there is change in the sequence of these nitrogen bases, it will lead to the change in the meaning of the code. And so we call that shifting of the language as mutation. So the genetic factor is encoded, is inherent in the sequence of nitrogen bases on the DNA, stretch of a DNA. What are the epigenetics? There are other factors. In older times, till a few years back or a few decades back, the epigenetic was restricted to the environmental impacts upon the genetic factors leading to the expression of genes. But now we have seen that there are many other factors also. That is, the expression of a gene cannot only be changed by the change in the sequence of nucleotides on that gene, but it can also be changed, or if not changed, you can say it can be modulated by other factors too, such as addition of another groups, methyl group, acetyl group. So methylation, acetylation, ubiquitination, submylation. So there are many factors which may impinge upon the DNA, upon the gene, to ask it to behave in a particular way. That is, its expression may be promoted or the function may be suppressed. So these factors, which are not directly genetically related, that is, they are not inherent in the sequence of nucleotides on the DNA, are called epigenetic factors. So epigenetics literally means above and on top of genetics. Modification in the DNA which changes its expression without, mind you, without changing in the sequence of nucleotides of that particular DNA segment. So the nucleotide sequence remains the same. The DNA remains the same. Still, its function is affected. Its function may be modulated. It may be induced to express itself, or it may be suppressed altogether. And those factors which act over and above this genetic composition of DNA are called epigenetic factors. These modifications do not change the DNA sequence, but instead they affect how cells read genes by altering the physical structure of DNA. So they change the reading pattern, such as methylation, acetylation, ubiquitination, etc., which may lead to loosening or tightening of the DNA wrap. You must have seen that when the DNA is being replicated or it is being transcribed, what happens? The DNA is loosened. DNA is in normal circumstances, the DNA is wrapped around the histone core, tightly wrapped around the histone core in the nucleosome. But if a DNA has to be transcribed, that is, it has to synthesize mRNA. During its activity, it will have to be loosened. The double helix will have to be unfolded, and then the entry of enzyme will have to be facilitated. So if attachment of a particular group tightens the DNA badly, it will inhibit the expression of DNA, whereas if attachment of any other group or any other modification, you can say, loosens the DNA, it will facilitate the entry of the enzyme and it will facilitate the expression of that particular stretch of DNA. So which may lead to loosening or tightening of the DNA wrapped around the nucleosomes, thereby facilitating expression or depressing it. If it is tightened, its function will be depressed. If it is loosened, it will be induced to express itself. An epigenetic trait is a stably heritable phenotype resulting from change in a chromosome without alteration of DNA sequence. So epigenetic trait is a stable trait. It may be carried from one generation to the another generation. So it is stably heritable too. It results into a stably heritable phenotype, that is characteristic expression of genotype into a phenotype. So these traits act, these epigenetic factors, they act without alteration of sequence of DNA, but they are stable and they can very much be transferred from one generation to the another generations, so they are heritable too. Regarding the passage of epigenetic modification through generations, whether they are heritable or not, these may be erased at times of fertilization. So since you know that at the time of fertilization, that is gametic fusion of sperm and ovum, there is reshuffling of genes. So during that reshuffling, the epigenetic changes might be erased, but they may also be maintained even during the reshuffling, and those changes which are maintained during the process of fertilization, they are carried from one generation to another generation, and a specification of centromere by CENPA, that is centromeric protein A is also such a character which is transferred from one generation to the another generation. That is, it is a heritable character. So you must have got an idea about the epigenetics, and we are going back to the current topic, that is epigenetics of centromere. Epigenetic factors such as attachment of proteins to the centromeric DNA and pericentromeric chromatin specify centromere. So centromere is specified by attachment of proteins to the centromeric region as well as the pericentromeric region. Not only attachment of proteins, but also modification of histone molecules as well as attachment of some groups to the DNA too. But the major genetic epigenetic changes which have got influence upon the determination or the specification of centromere are the attachment of CENPA to a particular stretch of DNA. As we have seen that particular stretch of DNA, that is the centromeric DNA, have preponderance of tandemly repeated units, that is satellite DNA, and the monomers are 171, about 171 nucleotides, and they are classified in the alpha family of satellite DNA plus HSAT 2 and 3. So even if there is a stretch of satellite DNA, even if it is being flanked by the pericentromeric heterochromatin, if CNPEA, that is the centromeric protein A, does not get attached to that particular DNA, it will not function as a centromere. So the attachment of centromeric protein A to the satellite DNA, which is to act as the centromere, that is the centromeric DNA, specifies that particular stretch of DNA to act as centromere. Now, this centromeric protein A also crosses the barrier of fertilization, and we have seen that it is synthesized independently of the DNA replication, and most of the proteins are synthesized in the S phase of the cell cycle. You know that M, G1, S, G2. So gap 1 is the gap, and synthesis takes place during S phase or the synthesis phase. Most of the proteins are synthesized, but in human beings in particular, the centromeric protein A, that is CENPA, is synthesized during the G1 phase of the cell cycle, and it remains tightly bound to the DNA. It has also been seen that CNPEA acts as template for the synthesis of the CNPEA during the daughter cells. So we see here that human CNPEA assembly starts after anaphase and finishes before the end of the G1 phase. So in between the anaphase and G1 phase, its synthesis is completed. So it is very much present in this daughter cell even before that daughter cell enters into the synthetic phase of the cell cycle. So centromere identity is maintained in cells that exist. Not only that, not only in the case of dividing cell. A question was raised about this centromeric presence during cell cycle, in the cells which exits the cell cycle. I'm unable to figure out the name of that student, but someone has raised the question that what does it mean that it is maintained in the cells which exit the cell cycle? So the oocyte development in the human female began even while the embryo is inside the womb of the mother, and it remains arrested at a stage which is called dictate stage. And when the woman attains puberty, that is, she becomes hormonally primed by the sex hormones and the hypothalamus. Only then the further division takes place and the ovulation occurs. So altogether, a number of years, these cells remain outside the normal cell cycle, that they do not undergo in division, and they remain in an arrested state. So even if these cells, these developing oocyte mother cells or primary oocytes, they remain outside the cell cycle, that is, they do not enter the M phase of the cell cycle, they maintain the centromere A. So centromeric protein A. So it is the attachment of the centromeric protein A which is of paramount importance for the centromeric stretch of DNA to act as centromere, except in case of holocentric insects, which have altogether done away with the centromeric protein A or its other variant. But in most of the other animals, what happens? That there is centromeric protein A of one type or the other type, that is either, it will be better to call it collectively CEN-H3, that includes the variants of the centromeric protein A2. So these are these cells which do not normally enter into the cell cycle for a longer period of times, for a number of years, 10, 12 years, they do have the centromere also. They do have the centromeric protein A attached to their centromere also. So at least 3 important criteria are likely to be required for epigenetic centromere inheritance. First, the mark must be stable enough to survive through the key cell cycle steps. So it should not get detached from that particular chromosomal region or the particular stretch of the DNA during cell cycle. Centromeric protein A does not get detached. Then self-templating. As I have told you earlier, that the pre-existing chromatin-bound CNPA molecule acts as template. Then third, replication of centromeric protein must be tightly coupled to the cell cycle. So that replication is tightly coupled to the cell cycle, and it is preluded, it is preponed. That is, most of the proteins are synthesized during the S phase of the cell cycle, whereas CENPA is synthesized between anaphase and N of the G1 phase of the cell cycle. All centromeric CNPA remains centromere associated during DNA replication and the subsequent mitosis. So this criteria, that they remain attached to the stretch of DNA throughout the cell cycle, that is fulfilled by the CNPA. This extreme stability is consistent with the role of CNPA as an epigenetic marker maintaining centromere identity. So it remains tightly attached throughout the cell cycle, that it imparts the centromeric property to the stretch of centromeric DNA throughout the cell cycle. It doesn't get detached, and even when the daughter cell is produced, what happens? It is synthesized at the earliest between the anaphase and the end of the G1 phase. So the most important and most remarkable and the most profounding impact, epigenetic impact, is exerted by CENPA, that is centromeric protein A, and its different variants. So here you see the epigenetics in action. This is a case of shifting of the centromere of Icus, species Icus. So even if you don't take it as a centromere of Icus, just take the example. You may treat as ABCD, whatever you may like. Here, it has this satellite DNA and the centromeric protein is attached to the satellite DNA. So you have got the normal centronormal functioning centromere at this point of the chromosome at the stage A. What happens that due to the some reason or the other, this CENPA shifts from its original centromeric point. So you see here that satellite DNA is here and the centromere had shifted, CNPA had shifted below. Now, this point will act as centromere, though it does not have the characteristic satellite DNA, but the shifting of the CNPA from this point to this point has imparted centromeric property to this new point of the chromosome of the new stretch of the DNA at centromere. And so this centromere had been silenced, and this centromere is called neocentromere. Mind you, someone of you has raised a question about neocentromere and silent centromere, and she was having the idea that one of it was recessive or dominant gene. No, it is not recessive or dominant gene. Silent centromere, the centromere which was previously functioning at the centromere, but it had changed its function. Its functioning at centromere had been seized. And another point, the new point which functions at the centromere is called neocentromere. So what happens? Ultimately, this DNA, satellite DNA, it shifts to the point where the centromeric protein was attached. And so it had become a well-organized centromere called the neocentromere, and this point has lost its centromeric property. So you see that the stretch of DNA was similar here, but due to the shifting of this centromere, the centromeric protein A, the centromeric characteristic change. So this change in the characteristics of centromere, despite the sequence of nucleotides remaining the same due to the attachment of a particular protein, is called epigenetic specification of the centromere. So you can see for yourself that how this epigenetics works in case of changing the centromeric spot. And this changing of the centromeric spot in an individual may have catastrophic consequences, may lead to aneuploidy in that animal, that individual. It may lead to development of cancerous cells, and it may cause the death. But from the viewpoint of evolution, it has got importance because it may lead to the development of the sexual isolation and further speciation of the organism, as has happened in case of icus, and it had been widely studied in case of icus. Now, other epigenetic influences. A question was raised that how methylation, acetylation, or addition of another groups to the centromeric, to the histone proteins of the centromeric region creates an environment for epigenetic influence of the upon the centromeres. So how does that environment created? So I am dealing with that very question, that how methylation, acetylation, etc. will create an environment for the expression or suppression of a particular gene. As in case of the backgrounder, back to the basic slide, I have said that methylation, acetylation they may lead to the loosening or tightening of the DNA bound around the nucleosome core, and in that way they can have effect upon expression or suppression of the gene. Similar factors are here, I have detailed, and those factors are acetylation, addition of acetyl group, methylation, addition of methyl group, ubiquitination. One of the students had raised a question that what is ubiquitin? So it is a 76 amino acid regulatory protein, small regulatory protein. It has a regulatory role in the cell and plays role at many points. So due to its ubiquitous presence, present all at all places, ubiquitous present, it has been called ubiquitin. So ubiquitin is a small 76 amino acid proteins, and its addition and deletion to the DNA core, the nucleosome core, the histone proteins, can have impact upon the expression of the DNA. Either it may induce the expression of the DNA or may suppress the expression of the DNA. So these changes, that is acetylation, methylation, etc., alter the DNA accessibility to enzymes. So let us have a glimpse of the changes. You see here that here acetylation is taking place, methylation is taking place, ubiquitination is taking place, sumoylation is taking place. What is sumoylation? Sumoylation, sumo, a small ubiquitin-like modified protein. So this is not ubiquitin, but it is also a small protein which is similar to the ubiquitin and it modified. So it is called sumo, a small ubiquitin-like modifier protein. So addition of sumo to a particular structure chemical is called sumoylation. So you see here that this histone core and DNA is wrapped around it, and acetylation, methylation, ubiquitination, sumoylation, or phosphorylation may take place by adding these groups, and their addition will have impact on the expression or suppression of this DNA. Coming to this slide. So what happens that attachment of these groups will impact upon the accessibility of enzymes. So either they will make the enzyme more accessible or they will make the enzyme inaccessible. If they lead to the tight binding of the DNA, the entry of enzyme will become more difficult. So enzymes will be inaccessible, and without those enzymes, DNA cannot be expressed. But if the attachment of a group loosens the DNA, then the entry of enzymes will be facilitated, and so attachment of that group will lead to the expression of the DNA. So altering the pattern of DNA expression, changes alter DNA accessibility to the enzyme, altering the pattern of DNA expression. In a way, they create favorable environment. This was the question, that how they create favorable environment. They create favorable environment for functioning. If their attachment leads to the loosening of the DNA, they are creating favorable environment for the expression of the DNA. Whereas if their attachment leads to the tightening of the DNA, then they are creating favorable environment for the suppression of the DNA. So I think it is clear to you that how this favorable environment is being created for DNA functioning, favorable or unfavorable environment. For example, H3K9. H3 is the histone 3, K is the standard abbreviation for lysine, 9 is the ninth lysine from N terminal of the histone. So this is ninth lysine of histone 3. If it is acetylated, it is called H3K9AC. If it is acetylated, genes get turned on, whereas its methylation results in the silencing of the gene. So there is a slot. The slot is histone 3 number 9 amino acid, lysine amino acids. And now you can see that additions of two groups have diametrically opposite influence on the expression of the DNA. If K9 is acetylated, then DNA will be expressed. And if K9 is methylated, then the expression will be inhibited. So the addition of this group or that group is creating favorable or unfavorable environment as far as the expression of the gene is concerned. In a regional centromere in organisms such as humans, mice, flies, etc., canonical H3 histones are dimethylated at H3KME2. So H3 is histone 3, K is standard abbreviation for lysine, 4 is fourth amino acid from the N terminal of H3 protein, and Me2, it has been methylated twice. So it had been dimethylated. So list 4, that is like 4, has been dimethylated, which facilitates incorporation of CNPA and thus plays a key role in organization of centromere. Now, you can see that how these roles are intertwined, interdigitated around each other. Very interesting. You see, I have told you earlier that it is the attachment of CENPA to a particular stretch of DNA that imparts centromeric property on that particular stretch of DNA. But some other factors are facilitating the entry of CENPA to that stretch of DNA, and that is methylation of fourth lysine of canonical H3. So if it becomes dimethylated, it facilitates the incorporation of CNPA. So CNPA was having epigenetic influence on the development of centromere, and since this H3K4ME2 is facilitating the CNPA, it is facilitating that epigenetic influence. So epigenetic influence that we see apparently is the attachment of CNPA, but this attachment is being facilitated by H3K4ME2. That is, H3K4ME2 is acting as an epigenetic influence on the facilitation of another epigenetic influence, that is the entry of the CNPA. So in other words, you can say that it is creating H3K4ME2, that is canonical histone 3, methylated at lysine 4, dimethylated. 2 methyl groups attached at lysine 4, dimethylated K4H3. It is acting as an epigenetic factor for entry of the another epigenetic factor, that is CNPA. So it is facilitating, it is creating a favorable environment for incorporation of CNPA to the stretch of DNA, which will have to act as the centromere. So it plays key role in organization of centromere. Any depletion of H3K4ME2 has been shown to result in the failed incorporation of CNPA. If this H3K4ME2 is depleted, if its level goes down, then it results into the failure of CNPA attachment to the centromeric stretch of DNA, and it will ultimately lead to the malfunctioning, to the silencing of that centromere. Centromeric histones lack acetylation. So you see here that acetylation will facilitate expression of genes, and methylation will facilitate suppression of genes. So centromeric histone lack acetylation. That is, these genes are not expressed in a big way. The hypoacetylation of histones and methylation of DNA at the centromere alter the chemical interaction between histone and DNA and define a region distinct from euchromatin, traditional euchromatin and heterochromatin. These modifications maintain a heterochromatic nature of centromeric chromatin. So centromeric chromatin remains tightly bound to the DNA, and it is the heterochromatin which remains tightly bound and it is not expressed. Heterochromatin may be facultative or constitutive. The constitutive heterochromatin remains always tightly bound and they are not expressed, whereas the facultative heterochromatin may remain tightly bound in some phase and it may be expressed, loosened in another phase. So here the centromeric DNA, due to the attachment of, due to hypoacetylation, that due to lack of the acetylation or low layer of the acetylation, low level of acetylation and methylation. You have seen earlier that acetylation will promote expression of DNA, methylation will lead to suppression of DNA. So here, on one hand, the centromeric DNA is hypoacetylated, that is, its expression had been inhibited due to lack of attachment of acetyl group, and as well as it is methylated too, and methylation will further suppress the expression, and so it maintains these two specifications. They maintain heterochromatic nature of centromeric chromatin. Now, the pericentromatic chromatin, that is the chromatin flanking the centromeric region. You have seen in the earlier slides that there is a centromeric region in the middle and flanked by the pericentromatic region. The chromatin flanking the centromeric region are called pericentromatic chromatin. So they are also defined by methylation. For example, H3 and H4 in heterochromatic regions surrounding the centromere can be methylated at list line H3 K9 Me2. Here in the centromeric region, it was H3 K4 Me2, and here it is K9. That is the 9th lysine from the N terminal is dimethylated, and the 9th lysine also get trimethylated. So dimethylation of list 9 in canonical H3 and in canonical H4, it is the list 20, that is the 20th lysine remains trimethylated. So these methylations of H3 and H4 lysine molecules, it has got an imprint on the heterochromatic regions, and these modifications serve to recruit proteins such as cohesin. So these methylations lead to recruitment of proteins such as cohesin. Cohesin is the protein which keeps the two sister chromatids attached to each other, and that is why at primary constriction at the level of the centromere, the two sister chromatins remain attached to each other. In the diagram we have seen in the third slide or the fourth slide of the first part of the lecture on the centromere and segregation fidelity. So the modifications in the pericentric chromatin leads to the recruitment of cohesin, and this cohesin has adhesive impact on the states to maintain a structure of pericentric chromatin. Loss of heterochromatic modification is thought to result in aberrant centromeric fraction. So if these changes do not take place in the pericentric centromeric chromatin, the centromere will function aberrantly, and that function will not lead to the segregation, the fidelity of segregation, that is the faithful segregation, will not be possible then, and it will result in presence of cancer cells which are characterized by high degree of aneuploidy. So here you have one epigenetic factor of paramount importance, that was CENPA, but you see here that methylation will also facilitate the entry of the CNPA, that is methylation will create favorable environment for the entry of CNPA at the centromeric stretch of DNA, and that's why this methylation will ultimately create favorable environment for the formation of the centromere. And the pericentric chromatins, modifications in the pericentromeric chromatins are also of importance, because those modifications in H3 at lysine 9 and H4 at 20, they act for recruitment of cohesin, which is important for keeping two sister chromatids together. So if these methylations do not take place, cohesin will not be recruited, and these two sister chromatins will not be brought together. They cannot be arranged biradially on the equator, and ultimately they will be segregated in an anomalous way, leading to the aneuploidy, which may result into the cancer and other diseases. So here you have seen methylation, acetylation. Now, chromatin modifications, you can have an idea that how chromatin modifications, that is epigenetic factors, act upon the expression of genes. So you see here acetylation. Acetylation will lead to, in H3, H4, etc. In all the cases, acetylation is leading to transcriptional activation. I have told you earlier that if acetylation takes place, it will lead to the expression of genes. So in all cases, whether it is taking place in H3, H4, H2A or H2B, it is leading to transcriptional activation. Phosphorylation, which also lead to transcriptional activation. Methylation here is leading to transcriptional activation, where in 1723 and H3, whereas methylation here in this case, it will lead to transcriptional activation if methylation has taken place in H3, 4, 36 and 79 amino acids, but it will also lead to transcriptional repression. So methylation will have both impacts, but acetylation and phosphorylation, they will have only positive impact on the expression of genes. Ubiquitination, transcription activation and repression, it may lead to both expression if it is attached, if ubiquitination is done at 123 and 120 amino acid, it will lead to transcription activation. If 129 is ubiquitinated, then transcription repression. Similarly, sumolization will lead to transcription repression. So you can have an idea, broad idea, that this table is not that important from the point of centromere, but the question was how favorable environment is being created by the methylation, acetylation, sumolation, or ubiquitination. So this table gives you a broad idea that attachment of this group to that particular amino acid will lead to the suppression, or to attachment of that particular amino acid, that particular group to that particular amino acids will lead to suppression. So these, despite the DNA remaining the same, despite the sequence of nucleotides on the DNA remaining the same, the attachment or detachment of these factors on this or that amino acid may lead to the promotion of expression or suppression of the expression of the genes. So they have got epigenetic influence on the gene expression. So that was all about the expression of genes, that the epigenetic influence upon the expression of genes as a broader idea and a pointed idea about how centromere is affected by the epigenetic factors. So now we can have an idea that how centromere was formed, specified by the attachment of the centromeric protein A, how centromeric protein A differs from the canonical nucleosomes, that is the nucleosomes, that is the canonical histones, that is the histones found at another sites of the DNA, and what are the differences, how they lead attachment of the CNPA lead to compaction of the nucleosomes, and that provides integrity to the centromere. And then in the next class, in the last class, we have seen the structural modification in the DNA, in the centromere, that how a point was acting as a centromere, which led to the attachment of only one spindle fiber microtubule to that point in case of budding yeast, but a region was acting in case of fission yeast, and a more broader region was acting as centromere in case of human beings. And that was characterized by the presence of tandemly repeated satellite DNAs. And we have seen that the satellite DNAs are, which were earlier regarded as the junk DNA, have now been recrystallined as the multitasking apparatus of our genome. Now, in today's class, we have seen that epigenetics is different from genetics. That is, without changing the sequence of nucleotides on the DNA, we can modulate the functioning of DNA. The DNA functioning may be modulated in the nature, or it may be modulated in vitro, and that modulation by non-DNA factors, non-genetic factors, are called epigenetic modifications. And the epigenetic modifications may have profound impact on the functioning of centromere formation, because you have seen that how a centromere is silenced and how a new region of the DNA acts as a centromere. So we have seen that how acetylation promotes expression, methylation suppresses expression of the genes, and how interaction between these two will lead to expression of a particular gene or suppression of the particular gene. And we have also seen that the stretch of DNA in the centromere was not only hypoacetylated, but it was also methylated. And this hypoacetylation as well as methylation has changed its characteristic to heterochromatic region. That is, it is not expressed, it remains tightly bound. And the methylation also promotes the entry of the centromeric protein A, and it, in that way, it facilitates the epigenetic specification of centromere. So that was all about the epigenetic influences of centromere in today's class. The next class will be concerned with the process which governs the fidelity of segregation, that how the cell controls the segregation through the centromere, and how the kinetochore attached to the centromere halts the process of segregation if the attachment is not perfect, and how it proceeds when the attachment is perfect between the sister chromatids attached on the equator. So the last part of this lecture is concerned with the segregation fidelity, which is maintained by the centromere. So in the next class, which is to be held tomorrow, we will see that how the centromere maintains the fidelity of segregation. Thank you all for being with me. Thank you.